DNA, and then you transform this and then you introduce this DNA segment into the plant genome. Okay, so when you introduce this, it will stop the plant, okay, from transcribing a gene into mRNA. Okay, so bila you stop transcription of the gene, okay, in the plant yang you transform to, you will prevent the process of translation. Okay, so when there is no translation, there is no gene product lah. Okay, so this is what we call as knockout mutant. Macam kalau you tengok Gusti kan, knockdown kan, macam knockdown, knockout macam itulah. Okay, so we call it knockout mutant because the ability to make the gene product is actually being removed lah. Sekarang the plant cannot make the gene product. Okay, because of the insertion of this DNA into the genome. Okay, alright. So, however, the insertion is actually random. Dia bukan specific sites of the genome. It can go into different site of the genome. Okay, can result in the gene being merely altered. Okay, kalau dia random, maybe the gene can be also produced because they're random kan. So the gene can still make protein, but it will be deficient or work differently than the wild type. Alright. Okay, kalau if you have any question, let me know. I will try to answer. Okay, like I said, one of the uses of mutants are to study gene function. Okay, so basically, when scientists disrupt a gene, okay, alright, when you disrupt a gene, we hypothesize that the phenotype, okay, or the trait or the characteristics of the plant will be different, okay, from the wild type, okay, or from the normal unaltered plant of the same variety. So, for example, if the mutant show asymmetrically shaped leaf, okay, so bila you create the mutant tu dia punya shape of the leaf tak jadi symmetry lah. Okay, so we can deduce that the normal function of the gene is actually to ensure equal growth on both sides of the leaf mid-vein. Okay, so without the properly functioning gene, the leaf developmental is abnormal. Okay, so macam itulah. Bila kita mutate that gene, okay, tiba-tiba dia jadi shape leaf yang different. So we know that we can actually hypothesize or deduce that the gene involved in the growth of the leaf mid-vein. Okay, faham ke? Boleh ke? Okay. Alright, so to prove that the gene is actually responsible for the leaf shape tadi tu, okay, now we introduce back, okay, the complete sequence of the gene. The scientists would carry what we call as complementation experiment. Okay, so this complementation experiment where we introduce the wild type copy, okay, or the form of the gene yang complete, okay, introduced to the mutant. Okay, so we expect that when we introduce to the mutant, okay, so dia akan jadi normal. Okay, alright. So the technique to introduce the wild type copy tu sama lah teknik yang kita guna masa kita nak mutate the gene tu. Okay, faham ? Okay, so if the transgenic or the genetically engineered progeny plants of the transformed mutant plant have normal leaf shape, then we can confirm actually the introduced gene is the same as the gene that was disrupted in the mutant plant. Okay, so this method to figure out functions of gene by knocking out specific genes and looking for deviation from the wild type, kita nak tengok apa dia berlainan daripada wild type, this is what we call as reverse genetics. Okay, I think I dah mention last week lah what the difference between reverse genetic and forward genetics lah. Okay, so in reverse genetic, okay, we mutate the gene, lepas tu kita tengok what is the function of the gene. Gene tu cause apa? Okay, change of the shape of the leaf, mungkin the change of the height of the plant, okay. So this is what we call as reverse genetic. Okay, why we said reverse? Because the scientist already knows the sequence of the gene. Kita dah tahu the sequence, tapi we want to find out what is the function of the gene. Okay, so basically we work backward, okay, from how geneticists work before the days of genome sequencing. So we be able to know the sequence of the gene because of the advancement in the genome sequencing lah. Okay, so the opposite of the reverse genetic is actually what we call as forward genetics lah. Okay, so in forward genetics, kita mutagenize the population of plant. Okay, we create disruption randomly throughout the genome. Okay, and then kita tengok this effectively lead to many different phenotypes lah. Okay, so in forward genetics, the scientists choose a mutant plant with a phenotype. Okay, kita nampak ada phenotype yang different, dan kita try to figure out the gene responsible for the phenotype. Okay, so this technique of forward genetics is more time consuming lah, because it involves process what we call as mapping. Okay, so where we try to create a map, treasure map, without knowing anything about the treasure. Okay, kita ada mutant, lepas tu kita dapat phenotype, tapi kita tak tahu apa gene yang responsible for the phenotype. Okay, so this analogy, in this analogy, the gene responsible for the loss function is the treasure. Okay, kita kena buat map dia. Okay, so we rely on specific sequences of DNA called markers to narrow down the location within the genome. Okay, so the next lecture kita akan belajar about molecular markers lah. Alright. Okay, so to give you clearer idea on what is forward genetic and what is reverse genetics. Okay, so this is the diagram lah. Okay, so forward genetics, as mentioned previously, is actually we initiated the study to determine the genetics that underpinnings the phenotype variation. Okay, so we try to determine, kita nampak phenotype dia. Lepas tu kita try to determine what is the gene yang responsible for that phenotype. Okay, so in many cases, the observable variation has been induced using a DNA damaging agent, mutagen. Okay, but also can happen naturally lah. Okay, so the investigator eventually ends up sequencing the genes or genes thought to be involved in that phenotype. Okay, so for example, you create mutants. Okay, so you nampak satu yang different. Okay, so you try to figure out the gene responsible for this phenotype. Okay, so this is what we call as forward genetics. Okay, and then we have reverse genetic, where we know the sequence. Okay, so this is the other way around. Okay, but we don't know the function of that gene is uncertain. Okay, so basically we disrupt the gene yang kita tahu the sequence tu, or modify, then kita tengok apa phenotype dia. Okay, so we know the genes, so kita induce mutagenesis, and then kita observe the phenotype. Okay, macam for example, kita nampak the plant dwarf become dwarf, so kita tahu, oh, the gene related to height, okay, for example. Alright, so this is just summary, okay, of between the difference between forward genetics, okay, and also reverse genetics lah. Okay, so forward genetics, the origin is the phenotype. Okay, so we know the mutant phenotype, okay, and we have the mutant population. But we want to know what is the gene responsible for the phenotype. Okay, so what they do to figure out the gene responsible for the phenotype, you can do genome sequencing. Okay, you can do transcriptomy. You can use the technique of map-based cloning. Okay, and then you can use the technique of microarray of phenomics. Okay, then you know at the end, based on the chosen techniques, okay, where you want to know the gene responsible, okay, then the final destination will be you know the gene function of that mutant. Okay, all right, so this is different with reverse genetic, where the origin is actually, we know the function of the gene, okay, but you don't know the phenotype. Okay, what is the gene responsible for? Okay, so you create mutants. Okay, then you think by using this technique, okay, to create mutants. Okay, so you validate the phenotype. Okay, so what you know at the end, you know the phenotype. Okay, what is the gene function of the gene function that you mutate just now. Okay, all right. So any question? Tak ada?
Tak ada.
Okay, semua okay? All right, okay. I think quite clear. All right, so this is just basic lah supaya you nampak between the wild type. Okay, this is one example lah, mutation that caused a single change to a nucleotide. Okay, so on the left is actually the wild type allele of a gene. Okay, you know that there is a sequence in the DNA GAG. Okay, so then it being transcribed. Okay, so dia akan hasilkan mRNA, and then you get the polypeptide. Okay, so but if there is mutation, okay, so then you perasan bila mutation, okay, it can be induced or it can be happen naturally. Okay, for example, in this the mutant gene is actually the nucleotide is just one change of nucleotide, you can say, daripada G dia change kepada A. Okay, so bila dia transcription, dia dah jadi different, AAG, and initially in the wild type, this sequence of codon produce glutamine. Okay, so but bila dalam mutants, even single nucleotide change, it cause the changes to the amino acid. In the mutant, they produce lysine. Okay, so this is the inactive mutant gene product lah. Okay, so this is to show you the effect of mutation. Okay. Okay, any question? Tak ada? Alright.
No.
Okay, clear? Okay, so so the role of reverse genetic is actually in this case we want to study gene function. Like I said, what is the function of the gene? This gene responsible for which phenotype? Okay, which characteristic or which trait? Okay, so the process of disruption or alteration can either be targeted specifically. Okay, you can specifically target dekat genome of the plant, kat mana you want to disrupt, okay, nak ganggu or nak mutate. Okay, this can happen if you use the technique of gene silencing, which is very specific. You can silence that gene. Okay, you can use the homologous recombination, or also you can use the technique of which is random disruption. Okay, where for example chemical mutagenesis is random. Bila you exposed to for example EMS, the mutation be random in the genome. Okay, but gene silencing very specific. So you boleh target dekat mana you want to make the changes or want to disrupt. Okay, and transposon also are something random. Okay, so bila you dah do the mutagenesis, mutation, then you have to screen the population. Okay, and to find out where is the lesion, kat mana mutation to occur. Okay, alright. Okay, so first the technique yang kita use for reverse genetic is what we call as chemical mutagenesis lah. Okay, where you use chemical, okay, to generate population of mutants. Okay, untuk forward genetics pun kita guna lah. Okay, long time ago untuk guna chemical. Okay, alright. So chemical mutagenesis ni normally it cause a point mutation, maknanya dia change pada nucleotide. Okay, generally are less deleterious than large rearrangement, and so high degree of saturation, okay, can be achieved in a mutant population using chemicals that generate single base pair changes lah. Okay, so or small insertion and deletion. Okay, so the two chemicals to cause that can cause changes to single base pair, okay, which is EMS, which is ethyl methane sulfonate, and also ethyl nitrosourea lah, ENU. Okay, so these chemicals cause changes to single pair base pair. Okay, while many reverse genetic techniques provide only loss of function alleles, a chemical mutagenesis can result in either loss of function, reduction of function, or gain of function of phenotype. Okay, so changes to the sequence tu boleh loss of function, maknanya plant tu tak the function of that certain gene tu totally lost, or you can reduce the function, or you can actually gain of function. Okay, using chemical mutagenesis. Alright. So, but the difficulty using point mutation for reverse genetic screening is that there are few cost-effective ways of screening. Okay, screening yang you want to screen the population of mutants from mutagenesis using chemical is actually quite expensive. And because for ways of you have to screen the population lah. Alright. So, but now we have technique, okay, what we call as stealing, which is targeted induced local lesions in genomes. We have the technique of NGS, new generation sequencing, and we have high resolution melting, HRM analysis, okay, where using this technique, you can screen the large population, okay, at a reasonable cost, okay, within an acceptable time frame, okay. So sekarang kita ada, sorry, saya tersalah, next generation sequencing, saya betulkan. And then you boleh use this technique lah to screen the mutants in the population. Okay, so Tilling, for example, use a variety of techniques for creating mutant population and to screen them, which actually employs mismatch specific endonuclease to identify the point mutation. Okay, so point mutation ni maknanya one single base pair lah. Okay, so using Tilling ni, okay, this endonuclease can cut, okay, is an enzyme, okay, to cut the mismatched part of the gene yang you dah disrupt tadi tu. Okay, so basically, when you want to screen the population of mutants, what you do is you grow the M1 generation, okay, and then the progeny of this plant, okay, which is what now we call the M2, we use for screening. Okay, so why we use the M2? Okay, because we want to identify the mutation that we identify in this process are actually heritable. Okay, maknanya diwarisi lah. Okay, and we want to eliminate the background somatic mutation that may be present in the M1 generation. Okay, that's why we screen the M2 generation, bukan M1. So you collect the seed from the M2 plants and you extract the DNA, and the DNA from this mutagenized individual is pulled, okay, then using the technique of PCR, okay, you try to amplify the gene of interest tadi, gene yang you dah disrupt tadi tu. Okay, so then the PCR product, you denature and then you allow to randomly re-anneal before you digest with celery juice extract. Okay, so you can identify the mismatch or the mutation in the amplicons in the PCR product when mutant and the wild type strand of DNA are re-annealed together, okay, to form a heteroduplex. Okay, so then these heteroduplex become target by the action of this enzyme. Okay, so only the samples yang ada mismatch, yang ada mutation are cleaved. Okay, faham eh? So kalau tak ada mutation, tak ada mismatch, dia tak akan cleave. So yang only yang ada mutants lah dia akan cleave. Okay, and then you can detect by using DNA separation technology. Okay, but the drawbacks of this stealing, techniques, it requires the construction of mutagenized population. Okay, but to have this mutagenized population is actually the process is challenging. Okay, and the techniques is also labor intensive, relatively expensive, and requires a high rate of mutagenesis to make the effort cost effective. Okay, so ni lah contohnya, kalau you expose the plant to mutagen of EMS, initially dalam wild type dia punya nucleotide is guanine. Okay, nanti you dapat O6 ethyl guanine. The guanine is being the addition of ethyl group. So nanti dia dia akan base pair dengan thymine. Okay, kita tahu guanine sepatutnya base pair dengan apa?
Cysteine.
Apa dia?
Cysteine.
Dengan cytosine. Okay, guanine sepatutnya base pair dengan cytosine, tapi bila you expose to mutagen, okay, jadi mutant, ada changes to the addition of ethyl guanine. So instead of guanine to base pair dengan cytosine, dia base pair dengan thymine. Okay, so this is the effect of mutagen lah. Alright. Okay, so this is to give you some example. Okay, for example, in the wild type, okay, cytosine ni bila dia exposed to the nitrous acid, which is also mutagen, okay, so the process of deamination occurs. Okay, dia jadi uracil. Okay, alright. So uracil sekarang ni akan base pair dengan adenine pula. Okay, so this is the mutant. Okay, alright. Any question? Tak ada? Okay. Lagi satu ni cytosine initially were exposed to mutagen of hydroxylamine, or the process of hydration occurs. So the cytosine become hydroxyaminocytosine instead of base pair with guanine. Okay, sekarang this modified base pair dengan adenine. Okay, so it causes the changes lah. Alright, so this is the example. Alright. Okay, so bila you for chemical mutagenesis, okay, when you want to screen the mutants, these are the technique lah. Okay, first you do the mutagenesis process by treating the seed or the callus of the plant that you want to create mutants using chemical EMS or ENU. Then you grow the M1 plants, the first generation. Then instead of screen the M1 population, like I said, you grow the M1 plants, okay, to get the M2 generation, then you collect the seeds. Okay, so you look for the phenotypes, okay, tengok apa changes yang happen to the phenotype or the trait, or you extract the DNA, okay, you extract DNA, then you screen the changes, dekat mana the target gene tu, okay, what happened, mismatch ke, single base pair apa yang change, using the T-link and GS or HRM. Okay, so dia boleh detect the changes to occurs kat mana. Okay, base nucleotide apa yang change. Okay, alright. Any question? Masih boleh follow ke? Boleh.
Boleh.
Okay, not too much Alright. Okay, alhamdulillah. Okay, so that is chemical mutagenesis. Okay, yang first tadi tu. Okay, so the second one is what we call as insertional mutagenesis. Daripada perkataan tu, you know, insert, you masukkan something, a segment of DNA into the genome of the plant yang you want to create mutant. So bila you masukkan DNA segment, dia akan ganggu the genome. Okay, so it will cause the mutation lah sebab you masukkan ada satu part, you insert. Okay, alright. So one of the most established method for reverse genetic is actually production of population of individuals that have insertion that disrupt the gene function at unique sites of their genome, in their genome. Okay, so the advantage of insertional mutagenesis is that individuals carrying an insertion of non-sequence in a specific gene can be identified using in a population using PCR. Which this technique PCR ni simple lah and also tak expensive. Okay, it's quite cheap lah. PCR ni common technique lah. Okay, so what you do, you pernah belajar PCR ke? Ada orang ajar you PCR tak? Belum lagi ke? Dah kan? Belum? Anyone tahu apa PCR? Tahu ke? Tak tahu. Ada siapa tahu PCR? Polymerase chain reaction? Tak ada?
Yang macam guna untuk detect COVID tu kan, Doktor?
Detect COVID. Okay, so bila you do detect COVID, okay, sebenarnya kita extract you punya DNA lah kan. Bila kita buat swab tu kan, kita ambil you punya DNA kan. So, jadi dalam DNA you tu kalau you ada COVID kan, okay, so you imagine kalau you ada DNA, COVID ada DNA dia tu, so jadi nanti dia ada design primer. Primer ni adalah primer yang primer ni being design adalah sequence yang similar to DNA of the virus, the COVID lah. Okay, so kalau dia match, primer ni akan bind to your DNA. Okay, so kalau you tak ada DNA virus tu, dia takkan bind. Okay, sequence. Tapi kalau you ada, dia akan bind, dia akan amplify. Jadi you akan dapat result, oh PCR positif. Maknanya dia amplify DNA of that COVID ni lah, that virus. Tapi kalau tak ada, dia tak boleh amplify lah. Okay, alright. So sekarang saya rasa saya tunjuk gambar video lah PCR Saya tak plan pun nak tunjuk, tapi saya rasa baik awak, at least you tahulah. Okay, okay, this is polymerase chain reaction process there.
And you can see the kids, they are getting excited knowing that we are coming. They're running here. You look at that.
Okay, so you nampak kan sampel, okay, DNA and PCR. Okay, so sekarang in our context, katalah kita punya sampel is the mutants tadilah, mutants kan. Lepas tu you want to screen the population, okay, so you extract the DNA of the population tadilah. Okay, alright, imagine so the DNA of the mutants population lah, okay. So you buat PCR, okay, imagine So this is the machine yang kita use untuk run PCR lah, okay. Okay, so this is the target DNA. Okay, so kita nak target kalau ada lah kan, katakanlah kita nak tengok mismatch ada ke, so kita pakai kita design the primers, kita synthesize primers, okay. So kalau ada, dia akan bind to this, nanti you tunjuk tengok dia bind to the DNA. Okay, so this is the target DNA lah. Macam kita imagine kalau you ada apa ni ada COVID kan. So ni DNA ni lah. So Okay, so macam saya cakap tadi, kalau you, for example, salah sampel you tak ada COVID, this primer tak boleh bind lah, because dia primer yang kita design ni, dua primer ni kita add dalam tube tu adalah akan base pair macam dengan the DNA, template DNA itulah target region. Maknanya base pair maknanya the sequence of the primer, katakanlah ni T, dia should be A. Okay, katakanlah ni G, dia C. Faham? Kalau dia tak ada sequence tu, dia tak boleh bind. Jadi bila kita tak boleh bind, you tak ada amplification lah. So you punya PCR negatif COVID. Macam tu. Okay, but kalau you ada sequence ni, okay, the primer being designed, dia akan anneal, dia akan bind to the target sequence ni. Lepas tu dia akan amplify. Dia akan, kita akan add nucleotide. Nucleotide tu akan ditambah ikut sequence ni lah. Kalau sequence ni adalah G, dia akan C, dia akan add macam tu lah. Faham the process? Okay, so the process being repeated depends on berapa cycle you letak lah. Kalau you letak 40, dia akan amplify banyak lagi lah. Banyak lagi. Okay, so that is the process of PCR. So any question? Any question?
Tak ada.
Yeah. Okay, so back to our business. Okay. For our, if you want to screen the mutants, okay, using insertional mutation, the PCR application is performed. Macam yang saya kata, ada dua primer, satu kat depan, satu kat belakang. Okay, so sekarang the primer, satu primer tu kita kena design, the primer will bind to the host gene specific primer. Okay, the host gene, host tu yang plant tu lah. And another primer, like satu primer tu, yang belakang tu kita kena design the vector based primer. So bila, ingat tak hari tu kita buat transformation agrobacterium tu, kita kan ada vector kan? Kan saya cakap, anything yang being inserted dekat tDNA region tu akan dimasukkan dalam plant. So satu lagi primer tu dia kena bind to the vector, to the tDNA tu. Okay, so kalau ada mutation dia akan amplifykan. Okay, thus amplification product will only be observed when the insertion is present close to the target gene. Okay, so from which the host primer was designed. Okay, so dia ada dua. Okay, dia akan berjaya amplify kalau ada mutation lah, when the insertion is close ni lah. Gene disruption using this technique result in normally total loss of gene function. Okay, macam kita cakap tadi lah, insertional mutagenesis different dengan chemical mutagenesis, because chemical mutagenesis can cause a loss of function, it can cause gain of function, it can cause reduction of function. Okay, so any question? Boleh follow lagi? Boleh ke?
Boleh.
Alright. So I hope that you fokus kat sini lah, jangan fokus study midterm pula Alright. Okay. And then insertional mutagenesis can also be used for activation tagging. Okay, nanti kita akan tunjuk lah, kita nama pun tagging, tag. Okay, the process of activation tagging is actually where you cause overexpression or atopic expression of a gene of interest. So, what in activation tagging, nanti sikit saya bagi contoh dekat arabidopsis later, is actually you ada construct. Imagine yang macam hari tu plasmid tu, kita ada construct. Okay, this construct ada strong promoter or enhancer element. Okay, so when introduced into the genome, this can be inserted into the genome of that plant yang kita nak mutate tu at random position. Okay, so katakanlah you target gene dekat dalam genome plant tu, target gene A yang you nak disrupt. Okay, so some of this insertion, bila you insertkan DNA segment tu, it will be upstream of the target gene of interest, so they akan enhance the transcription of that gene yang you target gene yang you nak mutate tadi tu. Okay, so again, the position of the insertion can be determined using PCR lah. Okay, so enhanced expression can create a phenotype even in a cases where loss of function mutation are not able to do so lah, because of redundancy of lethality. Okay, because kadang-kadang mutation ni cause lethality, mati, pokok tu mati. So you tak boleh nak study dia punya function. Okay. And then activation or disruption of gene function, the insertion can be detected from the T1 generation lah. Sekarang kita panggil T1 because transformation, and it can be easily followed in a population where the element will segregate in Mendelian fashion. Okay. Alright, so any question? Any question?
No.
Tak ada.
No, betul.
Tak ada. Okay, can I stop here? Okay, saya rasa you pergilah study untuk midterm awak tu. Okay, tapi boleh saya confirm tak awak punya petang ni dekat lab kat mana? Boleh ke? You tak tahu lagi lab kat mana petang ni?
Tak pasti lagi.
Tak pasti?
Dekat
Tapi saya tanya Dr. Latif lah sebab saya akan tengok juga awak ni lepas tu macam Sharifah hari tu awak tanya saya kan pasal result eksperimen sebelum ni kan? Okay, saya akan try to explain kat awak lah. Any question that you want to ask, nanti saya tanya Dr. Latif. Saya rasa saya stop dululah kat sini. Okay? Saya pun nak tengok-tengok untuk guta event yang plantation tu. Okay, saya stop kat sini dulu. Nanti kita continue another 2 weeks. Biasa minggu depan saya tak ada lecture kan. You all pergi free work kan? Betul kan? Hari Khamis. Okay. So lagi 2 minggu lah saya jumpa awak untuk lecture yang saya akan ambil pagi petang lah. Untuk to replace my lecture yang minggu depan boleh?
Okay, doktor.
Okay, so kita continue. Nanti saya confirm dengan Dr. Latif kat mana awak punya ni. Nanti kita jumpa petang lah, insyaAllah
Okay.
Alright, okay.
Alright.
Okay, alright, okay. Assalamualaikum.
Waalaikumsalam. Thank you, doktor.
Tapi minggu depan lagi 2 minggu kita start. Tapi lagi 2 minggu kita start. I hope that you dah baca my slide tu lah Boleh
boleh.
Alright, thank you. Assalamualaikum.